Hello, good evening. Our family, Biochemistry Simplified, Madam Winnie here. Thank you so much for the subscriptions, and I hope we've been able to get one or two which we didn't get right, but still we are here to learn. So today I'm going to introduce the polysaccharides. I had already posted on disaccharides, on the monosaccharides. So today I'm going to work on polysaccharides to finalize on carbohydrates as one of the biomolecules which we had to discuss in depth. So if you look at polysaccharides, from the name, it is a polymer of the monosaccharide units arranged in a linear order, or at the same time it could be having some branches. So today we are going to look at polysaccharides, which are polymers of the monosaccharides which we already learned. So to start with, let's look at the introduction, how we introduce the polysaccharides. From the name poly, it's a polymer of the sugars. That is the simple sugars which we saw. The components of the glucose are simple sugars, the fructose, as well as the galactose. So we are going to look at the polysaccharides, and they're defined as polymers of monosaccharides. So they're defined as they contain more than 10 or more monosaccharide units, but still they have to be joined by a glycosidic bond. Remember, I mentioned glycosidic bond at the time I was talking about this disaccharides. So when a glycosidic bond forms, there is always a water molecule which is released. So a glycosidic bond is still going to be involved because we're talking about the carbohydrates, but now it is a polymer of those monosaccharide units which are going to form the polysaccharides. So classification of these polysaccharides are basically two. We have the homopolysaccharides and the heteropolysaccharides. So the most commonly discussed ones are always the homopolysaccharides. And you look at the definition of the homopolysaccharides, they are made up of only one monosaccharide component. And if you talk about the homopolysaccharides, the best examples which we already know, we always have starch, you've already heard about glycogen, as well as cellulose. So we are going to start by looking at the homopolysaccharides, and later on in the last slides we'll be able to discuss about the heteropolysaccharides, of which the definition goes by hydrolysis will yield more than one type of monosaccharides. So let us dive into what homopolysaccharides are all about and the examples. The first one which you are used much with is starch. So if you look at starch, it is a form of glucose found in plants. And basically, if you talk about starch, the cereals, the tubers, all those are starch. So you talk about rice, talk about tubers like potatoes, cassava, all those are starch. So this starch, if you look at its structure, as much as it's a polymer of those alpha D glucose units, it is a length of many of those monosaccharide components in it. So it is a homopolysaccharide because the main component of the monosaccharide, the main component of the polysaccharide, which is the monosaccharide component, is the alpha D glucose, arranged in either a linear or as well as a branched chain. There are branches, or it's also linear. So based on the classification of starch, starch will exist in two forms. It's either an amylose or an amylopectin. So if you look at amylose, amylose is the linear part. So if we talk about the linear one, it means it doesn't have branches. It only has the alpha 1,4 glycosidic linkage. So we are talking about an alpha D glucose. Remember, there is a session where I discussed the cyclic form of those monosaccharide components, and we said the alpha D glucose, if you look at the glucose itself, by introduction or based on the naming of the monosaccharides, we found that glucose falls under the aldose as a functional group. It is a 6-carbon atom. So we were naming it as aldohexose. So when the aldohexose glucose is drawn in a cyclic form, it was going to form a 6-membered ring. So if you talk about an alpha D glucose, at carbon atom number 1 of the alpha D glucose, the OH is below the ring. And then now we are talking about D. So if you're looking at the linear form where we talk about the D and L, twisting your linear form in clockwise direction, we were able to know the position of the OH at carbon atom number 2, number 3, number 4, bearing in mind that at carbon atom number 5, the OH at carbon atom number 5 already form a hemiacetal with carbon atom number 1 having the aldehyde group. So here we are talking about the alpha D glucose. We are forming the 1,4, alpha 1,4 glycosidic linkage. It means at carbon atom number 1 of the first glucose residue and the carbon atom number 4 of the second glucose residue, then at carbon atom number 1 of the second glucose residue and the carbon atom number 4 of the third residue. So it's a polymer of alpha 1,4. So it means amylose is a type of starch, but then it only has the alpha 1,4 linkages or the alpha 1,4 bonds because it is linear. Only the 1,4 will represent the linear part, but then it comprises huge portion of starch is amylose, so 80% of it. The other type of starch is amylopectin. As much as it's homopolysaccharide, it contains only the alpha D glucose, but it has both alpha 1,4 and alpha 1,6. So it has the linear part, but at the branching part we have the alpha 1,6. So the alpha 1,6 will always come at the point of the branches, but the alpha 1,4 will represent the linear part as alpha 1,6 represent the branches. So it both has the alpha 1,4 and alpha 1,6. It is a polymer structure which is branched, but what we have to bear in mind, it is a homopolysaccharide, because on hydrolysis it will only yield one type of monosaccharide residue. So at this point in time we are talking about starch, and we have two types of starch, the amylose and amylopectin. Look at the structure of amylose, being that our alpha D glucose alpha, it means the OH at carbon atom number 1 was below, so it was easier to extend it downwards, and then it attaches itself to the 4th carbon atom of the second glucose residue, 1st carbon atom of the 2nd, 4th carbon atom of the 3rd, but it's easier to extend the glycosidic bond downwards because of the alpha D glucose. So it is a linear. The amylose is a linear type of starch, and then amylopectin, look at the linear part. Then at the branching point we have the alpha 1,6. So amylopectin is branched, amylose is linear. Another type of homopolysaccharide is cellulose. Cellulose is found in plants, so it forms the major structural part of the plants. But now it is also linear, just as amylose. The difference is amylose was alpha D glucose, but now cellulose is beta D glucose, forming a beta 1,4 glycosidic bond. So they share one similarity, that they are both linear. But the difference is amylose has alpha 1,4, but then cellulose has beta 1,4. Amylose, another difference is it has an alpha D glucose units. Cellulose has beta D glucose. For the cellulose, it means at carbon atom number 1, for beta, the OH is above the ring at carbon atom number 1. And that is how we determine the beta or alpha of a monosaccharide in cyclic form. So in this case, we have a beta D glucose. So it involves repeating disaccharide of cellulose, which is known as the cellobiose. The cellobiose is the repeating disaccharide in cellulose, and it is the one which involves 2 of the beta D glucose units, now forming a polymer. So animals always lack the enzyme to break down cellulose, but then this enzyme is always present in the bacteria of those ruminants, example the cows, and that is what allowed them to eat grass so that it can be digested. That is the structure of cellulose. You can see the beta, the OH is above, so it's easier to extend the beta 1,4 glycosidic bond upwards. Another one, glycogen. Glycogen is a component of homopolysaccharide which is found in animals. But glycogen is the similarity, there is similarity of glycogen with amylopectin because both of them are branched. But glycogen is more branched than amylopectin. So it means the similarity of glycogen and amylopectin is both of them have the alpha 1,4 and alpha 1,6 glycosidic bond. But the difference is amylopectin, storage form of glucose in plants, glycogen, storage form of glucose in animals. So let's see, it's a major glucose storage for the carbohydrates in animals as well as humans. It is similar to amylopectin, but it is more branched, meaning you can go 6 to 8 alpha D glucose units, then you meet a branching point in glycogen. But for amylopectin, you can go 20 to 30 glucose units is when you find a branching point. So it means that glycogen is more branched than amylopectin. Another difference is glycogen, the storage form of carbohydrates in animals or humans, but amylopectin, storage form of carbohydrates in plants. So glycogen is always stored in liver as well as the muscle cells. So the highly branched structure of glycogen will always permit the storage of that glycogen or glucose in a small space. So in that time, glycogen will always have stores because of its highly branched, so it permits glucose to be stored in form of glycogen, which is always released. When the glucose level is low, the glycogen stores will release glucose, which can be used by muscles during exercise. So we always see the difference between those two. Glycogen is highly branched than amylopectin. The structure of glycogen is there, so it shows that you go a few glucose units. I talked about 6 to 8 is when you can meet a branching point. It's trying to illustrate why glycogen is more branched than amylopectin, but the similarity is both of them are branched. So it shows the structure of glycogen and amylopectin. Here, down here, you can see how amylopectin structure looks like, how glycogen structure looks like. So you go close a few glucose units, then you meet a branching point in glycogen. But then when you compare with amylopectin, you'll have several glucose units is when you meet the branching point. So it tries to explain much that glycogen is more branched than amylopectin. We have other homo and heteropolysaccharides. So the first example talk about dextran, which is also homopolysaccharide, inulin and chitin, which are also examples of homopolysaccharides. And then you can see in dextran we have both 1,6 and 1,3 linkages. The 1,6 is forming the linear part, the 1,3 will form the branching part, but what you have to note here is it is a homopolysaccharide because on hydrolysis it gives us only glucose units. Here is the function of the homopolysaccharide. It is present in bacteria, used to maintain the plasma volume in clinical medicine, and lastly it relates with dental plaque because the bacteria, oral bacteria, will always be due to dextran, which is synthesized from sucrose. So the oral bacteria which is related to dental plaque is as a result of synthesizing dextran from sucrose. Inulin is also a homopolysaccharide which is composed only of fructose, but it forms a beta 1,2 glycosidic linkage which is present in the fructose units. It is the best one which is known much when you want to know how the kidney is in good condition. So we talk much about the clearance test. When you use inulin as one of the exogenous substances, it's used much in determining whether the kidney is in good state. Chitin, composed of N-acetylglucosamine. So glucosamine tells much. As much as there is an amine group, it is glucose. So the glycosidic bond is beta 1,4. It is also part of a structural polysaccharide in the invertebrates, for example, the lobsters, the crabs, as well as the insects. Then we have a class of heteropolysaccharide. We said heteropolysaccharide on hydrolysis, they yield more than one monosaccharide unit. So we have hyaluronic acid, HA, which has a repeating disaccharide of glucuronic acid together with N-acetylglucosamine. You can see the glucuronic together with N-acetylglucosamine will tell you how much it is an heteropolysaccharide. But then its function, it's present in the synovial fluid, it's also present in the skin, also present in the umbilical cord, and even the vitreous fluid. It's present there, and also it is produced by the mast cells. So the heparin itself is also one of the heteropolysaccharides, and we have several examples which talks much about being examples of the heterosaccharides because they are used much in medical applications as well as other functions which are explained on the table. So that one